Welcome everyone to today's webinar. This is a preview for the American Society of Human Genetics meeting in Vancouver, British Columbia. If you have questions, please type them in the questions pod or the questions section when you think of them. And if we cannot get to all of the questions, we will put them in a document and put them in the link for the specific webinar. The information for the webinar is in the link at the bottom of the screen. The slides from today's webinar and a PDF file are there now. As I said, this is a preview of the American Society of Human Genetics meeting, and NCBI will be an exhibitor at this time. My name is Bonnie Madak. I'm the moderator of today's webinar. We also have with us today 3 panelists, and then the other 4 names on the slide are the other NCBI staff that will be at the American Society of Human Genetics meeting. We will be exchanging the microphone as we go through the webinar, so there will be some pauses. I'm going to pass the microphone now to each of the panelists so that they can say hello and you can hear their voices.
Hi, this is Steve Sherry, NCBI.
Hi, this is Ben Busby, NCBI.
And this is Valerie Schneider, also NCBI.
Thank you very much, panelists. What I'm going to do is go through the general events at NCBI activities at ASHG, and then we will have each of our panelists talk a little bit about their individual presentations and then also have a conversation. This webinar is really designed for a conversation today. We do want to share the science with you, but I really wanted to get some information out to the attendees of the webinar as far as why NCBI exhibits, why we like to go to scientific meetings. One of the first things that will happen at ASHG this year is the HackSeq Genomics Hackathon. That will be occurring Saturday, October 15th through Monday, October 17th. Ben Busby will be talking about that a little bit more later in the webinar. The second workshop that NCBI is participating in is the Genome Resource Collaboration Consortium. I had that wrong. And the Genome in a Bottle workshop, that is Tuesday, October 18th. And then we have 3 different sets of posters that will be presented. You'll be seeing information about each of those posters in the course of this webinar. The NCBI booth will be open October 19th through the 21st. You will see those times at the end of the webinar. Our booth number is 521. And I've provided the link there for the abstracts and details. If you get the PowerPoint slides from the materials directory that I showed a few moments, and Ben, you may be already doing this, if you can retype that link, you'll need to get it separate, but we can put it in the question pod for those who want to get that link. We're also going to do a poll on this webinar because we're curious to see if it matters to attendees of scientific meetings as far as how you prefer to interact with NCBI staff. So the question is, at a scientific meeting, how would you prefer to interact with NCBI staff to learn about NCBI resources? Would you like to go to a workshop presentation, a poster session, see a software demonstration, listen to a scientific talk, or visit at the exhibit booth where you can ask an individual question, and you can have more than one answer on this poll. So I'll pause for a moment. We will talk about the results of the poll later in the webinar. I want to talk first about the human variation resources, that are available at NCBI. We have quite a few different databases. We have quite a few different tools. You can get to the variation resources page by going to NCBI. I have abbreviated it here on the slides with the bracket NCBI bracket. You need to replace that with www.ncbi.nlm.nih.gov/variation. I've also provided a link to the fact sheet, which describes the variation resources. These fact sheets are very useful and comprehensive about the various services, tools, and databases that we have. You can go to the fact sheets directory and then obtain any fact sheet that you want. One of the posters that will be available at ASHG is by Mike Fiola and his group, and it's about the dbGaP database. dbGaP contains genetic, molecular, and phenotypic data, and there are some other items of information there on the page. When you scroll down on the home page of dbGaP, you can see the latest studies that have been added to the database. And then you can also do an advanced search and have very specific facets of information, data fields that you are interested in. And you can get to the faceted search by looking on going to the link that is shown on your screen. Another poster that will be at ASHG is about the NGS, the Next Generation Sequencing Swift Cluster. This is a parallel computational framework to do large-scale genetic variation analysis in the cloud. And I did give Steve a heads up. Is there anything else you would like to comment on, Steve, for either of these posters?
Thank you, Bonnie. Yes, if we could go back to the previous slide, I've just mentioned for dbGaP here that this new advanced search is a demonstration of a powerful new technology we're deploying here at NCBI using solar faceting to permit specific compound searches of data sets or data descriptions. In this case, it's going to be the study description and content. And what you can do through selection of the check boxes is narrow down in simultaneous dimensions searches about concepts such as a particular disease area, a type of study design, a type of data, whether it's whole genome sequencing or genotypes or phenotypic information, and looking for a list of studies, or as you can see in these other grayed out tabs across the center, lists of variables, documents, or molecular data sets that are available that meet the study criteria. That list can then be either used to directly download data in dbGaP or to prepare a data access request, or possibly just to know that it's here and is connected to other public NCBI resources, and there might be aggregation on those data. So I think that's a useful, kind of exciting new technology that we're showcasing at ASHG and trying to put in production now. On Shunlin's talk, the next one, which is about the Swift analysis. This is variation analysis based on next-gen sequencing data that's been developed as a demonstration pipeline that works on controlled access data directly from dbGaP running in the cloud. In this case, it's Amazon. And it's following all of the security principles that are required for access to dbGaP data so that the data are encrypted in transmission and at rest. The SRI toolkit can read local copies of data pulled from dbGaP SRA into AWS and compute on it while leaving it encrypted at rest. And it has performance and accuracy properties that are comparable to other systems. So we think that this and some of the transmission speeds are superior to what you get if you're just doing bulk transmission of an entire data set up into, say, S3 and then trying to read the data from there. So this is something where there's going to be, I think, a lot of interest and growth in the community because data sets are getting larger. TopMed is going to be coming out and discussed at ASHG. That is the NHLBI flagship project that has 20,000 individuals in it in phase 1. So that might be something to be looking for both in dbGaP and as a scientific presentation on the just-in-time workshop. So I'll just stop there.
Thank you, Steve. The last poster that is related to variation resources is the ClinVar database, and that is by Melissa Landrum and her group. And that is being focused on both for researchers and medical practitioners alike. ClinVar is a repository for variants, again with phenotypic consequences and interpretations. You can see the variant descriptions in ClinVar, and it has more than 160,000 variations, which corresponds to more than 121,000 nucleotides. And almost 50,000 of those variations are indeed pathogenic, and they are also from more than 6,000 expert panels. So the time of each of those posters is on the slide, and you can also see that information on the conferences and presentations webpage that I will show you the link for at the end of the webinar. The next part that we want to talk about NCBI activities are hackathons. These are where a group of people get together to focus on a problem or a situation to come up with a solution. And I'm going to ask Ben to talk about this in more detail.
Hi, Bonnie. I'm really glad you asked about this because I love talking about hackathons. And I'm really very excited about HackSeek, the hackathon that is going on in Vancouver. So this is the largest NCBI-style hackathon ever. And there are going to be 10 teams working on 10 distinct software projects in Vancouver, and hopefully we'll get at least 8 or 9 working functional software projects. And you can go to hackseek.com, check out what the teams are doing. You can volunteer to help. There's social events that are going on around this hackathon, so that'll be pretty neat. And you can see the HackSeek front page here. I will be working not as an organizer, but with one particular team. I'll be leading a team working to auto-close bacterial genomes with long-read technology and potentially submit them to public databases. So that's something I'm very excited about. Additionally, after HackSeek, there'll be an NCBI-style hackathon at Cold Spring Harbor the following week, and then there will also be NCBI hackathons at NIH, definitely a biomedical informatics one in January, and then possibly another genomics hackathon in March.
So that's the activity for the hackathon both in Vancouver and subsequent weeks. And then Ben is also going to be presenting a poster about machine images. I'm going to also ask him to talk about this now.
So similar to the things that Chun Lin is interested in and what he's presenting, we have some other Amazon machine images that allow folks to do very powerful things with our software development kit. And one great example of this, I think, is being able to use GATK seamlessly with the SRA toolkit, basically to call variants directly from aligned SRA data sets. So that's something we're very excited about, and we'll be showing those machine images and possibly Docker images at ASHG. Also, the SRA toolkit works seamlessly with HiSat2, which is an alt-aware RNA-seq mapper, and then also an in-house RNA-seq map, or an RNA-seq mapper that we've built in-house based on BLAST called Magic BLAST, which is a product I'm very excited about, and in my personal opinion has a lot of possibility for being able to look at trans-splicing, possibly in cancer data sets. I also am very interested to show people some of the new things we're doing with eUtils, particularly its command line implementation in eDirect, as well as SRA Blast, which I think is an amazing technology because it allows anyone to get into the computational genomic space with public data sets very, very quickly.
Thank you, Ben. So for all of you who have a lot of data to analyze, using these machine images can help you save you from the need to have such large computational resources yourself, to do it in the cloud. Next, we're going to talk about the Human Reference Assembly and the Genome Reference Consortium. GRCH38 is one of those products that comes out of the GRC, and there will be a workshop on Tuesday, October 18th. I'm going to ask Valerie to speak about that now.
Thanks, Bonnie. I'm excited for the chance to tell folks about the GRC workshop that we're going to be having on Tuesday the 18th. And I just want to let folks know up front that if you're interested in coming, it's a free workshop, and you can actually sign up and let us know you're coming by visiting the GRC website, which is genomereference.org, and clicking on the link for the Eventbrite so we can get a head count. But the workshop is something that we've been hosting at ASHG for a number of years. NCBI is one of the members of the GRC, and we use this workshop as an opportunity to let folks know about the ongoing work on the reference assembly, as well as new features and new developments that are coming out not only in the reference assembly, but in the field of genomics and assembly building. And this year, for the second time, we're going to be partnered with the Genome in a Bottle Consortium, who's going to be spending some time talking about the development of infrastructure for reference materials that help let folks translate data from just genome sequencing into clinical practice. So on the GRC side of things in the workshop, we're going to be focusing on helping folks understand really the assembly model. So we encourage people who might be just entering the field of genomics for the first time, be they clinicians, basic researchers, or even educators, to come and just get a handle on what the reference assembly is all about. We'll talk about some of the challenges and the benefits of moving to the latest version of the reference assembly. We'll be talking about public resources at NCBI as well as other GRC centers that provide details about the reference assembly and tools that will help with data analysis. And again, we'll be talking about our ongoing curation efforts. We've got a number of speakers lined up. I'll be speaking about the background on the reference, but Tina Graves-Lindsay from MGI, another GRC member, will be talking about some new reference quality genomes that are being built for different populations. Jane Loveland from GenCode will be talking about GenCode annotation on the reference. And Deanna Church, who's at 10x Genomics, is going to be giving us all a preview about the emergence of diploid genome assemblies. And I'm sure Genome in a Bottle will have a great panel of speakers as well. So we really look forward to having people attend. So one of the other things I'll be presenting at ASHG is a poster on navigating GRC H38, the current reference assembly at NCBI. And I'm really going to be focusing on sort of expanding on what I'll be talking about in the workshop, current work that we're doing on the reference assembly, as well as the tools that we have at NCBI for taking advantage of it. One of the great things about NCBI being involved in the Genome Reference Consortium is that we're really sort of at the cutting edge of what's happening in the reference assembly, and we're building tools that allow users to take advantage of the reference and all of the features that come along with it, which are some features that other resources at other sites might not have. So I'll be talking about, as shown on the slide here, our genome remapping service, which allows folks to transition from older assembly versions to newer ones, as well as actually to map data between our alternate loci and the primary assembly of the reference. And I'm also going to be talking about a number of browsers at NCBI, including our newest browser, the Genome Data Viewer, with really emphasizing the features of this browser that let you take advantage of things like alternate loci and patches and navigating around the reference assembly as a whole. So I'm hoping to gather folks' interest in our new tools, and I'm really looking forward to getting feedback on the resources that we have at NCBI and how we can make them better.
Thank you, Valerie. Sorry I didn't put that on the screen when you were still talking about the other tools and resources. Now I want to go into a conversation with our panelists, and this is where I really hope that the webinar attendees appreciate the wealth of experience and interest that we have with this panel today. When I have gone to exhibits in the past, serving in the NCBI booth, people walk down the aisle and they look and they say, well, why are you here? What does NCBI have to sell to me? And of course, we don't have anything to sell because we're federal government funding. Or people will say, well, why are you here? Everybody knows about NCBI. A few years ago at the Plant and Animal Genomes meeting, I was working in the booth and they would walk by and say, oh, I know everything about NCBI. I said, well, do you know about the new BLAST formatting service? And they said, what are you talking about? So they would step into the booth and then I would spend 15 to 20 minutes in conversation with them, showing how the new BLAST formatting was going to help them analyze their data. So I want to have our panel answer a few questions that I will pose to them and maybe have a conversation amongst themselves and show, of course, the homepage for NCBI. And if you click on the About NCBI link, you'll see some information. And also our mission. And I'm going to ask Steve the first question as far as how does NCBI achieve its mission by going to scientific meetings?
Yeah, so if you scroll down a little bit below what you're showing right here, this is being broadcast, that below basic research is the way NCBI carries out its missions in these bullet points. And I think that conventions and forums like ASHG are important because they are a very efficient way for NCBI scientists and supervisors to meet with a broad array of users, researchers, fellow data submitters, data users, and policymakers. Look for both scientific advice and user education, as you just mentioned, when people come to the booth and we want to show them services or improvements we've made. We also, as shown in some of these bullet points, have responsibilities for developing, supporting, and coordinating access to a variety of databases. That's one of the second to the last point. And we want to make sure the community has both an advisory role and an educated role to be a power user or find out if our interface, our presentation is suitable to the needs of the community. Science is always changing, and especially in human genetics and genomics, it's changing annually in very profound ways as new technologies come on. And so there is a challenge in both reinterpreting old data to keep it relevant and useful in new organizational paradigms. And I think that's true for a lot of human genetics, where we started with cytogenetic maps of disorders and we are now down to high-resolution maps. But there's many other areas in human genetics, especially around phenotypes, clinical definitions, standards for reporting, where NCBI uses meetings like ASHG as a way to reach out to professionals, both for research and clinical use.
Thanks, Steve. I'm going to ask both Valerie and Ben to share with us who they want to talk with at the ASHG meeting. We won't name any names, but at least to get a sense of the people that they want to talk with.
Thanks, Bonnie. Yeah, it's a great question. There are a number of folks that I'd be interested in talking to at ASHG this year, and they fall into a couple of broad categories. The first might be folks who are producing assemblies, and that would be people who are using newer sequencing or assembly technologies or producing diploid assemblies, as well as folks just new to the assembly building process. So maybe it's an individual lab that's trying to build a genome assembly for the first time. And also groups that are trying to make what they would consider reference quality assemblies. And for all these groups, I'm interested in how well NCBI is able to represent the assembly data that they're producing. I'd like to ask questions about whether we're capturing features of their assemblies or the raw data that are important. And I'm also interested in just sharing with them some of the new tools that we've been developing to help them assess assembly quality even prior to submission. And wearing my GRC hat, I'd be interested in understanding the data they're generating, if it's going to be public, and if it's something that we might be able to use in the reference assembly. I'd also love to talk to folks who are making the transition from GRC H37 to GRC H38 to learn about what's going well for them and what's not, finding out what tools they need to make this easier, and again, introducing them to some of our resources like our remapping service that can help, and explaining some of the features of GRC H38. And I guess the last category would be to talk to folks who are using browsers and genome viewers as part of their work and really give them an opportunity to see how our tools take advantage of the features of the reference assembly, show off some new display features that we have, and some resources that may be very specific to the types of work that they're doing. So I'd love to talk to any of these folks that are out there.
Well, as Valerie said, I'm very interested in talking to folks about viewing genomes, but I'm also very interested in computational genomics folks who interact with genome data on the command line. And I'm very interested in discussing discoverability with them in terms of finding the genomes and annotations that they're interested in, and submitting the genomes and annotations they're interested in, as well as using that data with tools, both tools in terms of tools we produce at NCBI, such as BLAST and many different iterations of BLAST that we now produce, as well as external tools for many domains. For example, SNP annotation. There are dozens of external tools, and we really want to know how well our data works in those external tools and what we can do to present our data to make it work better with those external tools. I also always look forward to talking to tool developers themselves, both in the sort of open source community, people that I'm personally a very big fan of, as well as people in the commercial community, some of whom make excellent tools. So that's something I certainly look forward to. Yeah, thank you.
Thanks, Ben. I know that Ben and Valerie will both have more to say in a few minutes. I'm going to ask them another question, but I'm going to next ask Steve, what are some of the changes that he has seen since he's been at NCBI regarding the science of human genetics, and whether he remembers any of those in connection with an ASHG meeting. I know that I remember many years ago, when imprinting was first discovered, we initially thought that both chromosomes from the mother and father were equally expressed, and found out that the phenomenon of imprinting meant that a certain gene from a certain parent was turned on preferentially. I still remember the human genetics meeting where that was the new discovery. The organizers of the meeting had to keep changing the meeting location to go to a bigger and bigger and bigger room, and they were still spilled out into the hallways such that the fire marshal had to come by and say that no one else could go into the room. So Steve, I'm going to ask you to just give us a reflection on some of the changes in the science of human genetics.
Thanks, Bonnie. I started at NCBI in 1998, and that was the year we started dbSNP. And at that time, there was no human genome sequence. The project was still in the middle. I guess we were sequencing clones, and there were a lot of them, but we hadn't done any kind of assembly yet. So at that time, I mean, since then, human genetics has seen, a profound evolution in terms of our understanding of the genetic information. We've gone from recombination and radiation hybrid-based mapping into physical maps where we can actually talk about alleles on long sequences of sequence. And so that becomes an organizing principle for everything in variation, and it becomes an organizing principle for traits and things that you want to connect, such as EQTLs and expression. So you can understate, how profound that is for genetics. It's like adopting the metric system or something that allows us to finally put all of this in a common cross-comparable system of coordinates. In the same 18 years, dbSNP has grown to be, right now, I looked this morning, it's 154 million variants, and that's primarily out of some community resource projects that were international in scope. NIH was a partner, NCBI was a partner as part of NIH, such as HapMap, 1,000 genomes, that produced a lot of data. Those are examples of meetings where some of the value of ASHG and its convening function, again, was that was a place we always had pre-meetings for these large workshops and consortia where we would be analyzing the data, having a face-to-face meeting, giving presentations, with 40, 50, 60 people, and then there was always going to be a plenary talk about the latest science coming out of these showcase projects. And like you were saying about imprinting, these were moments where it was the largest room would be filled up, or they didn't book the largest room and it was a room filled, overflowing, and the fire marshal was again saying, okay, move along, move along. And I think this coming year is going to be another example of that. TopMed, which is one of NIH's showcase medical cohort projects, will be reporting their phase 1 results at the late-breaking plenary session on Saturday morning. So that's another opportunity to go hear, some of the latest large-scale sequencing and analysis research that's coming out. ASHG is, always the convention where we try to get the word out to researchers of what NIH is doing.
Thank you, Steve. I'm going to ask Valerie and Ben to describe what they want to get out of attending the meeting. I know we've already heard who they want to talk to, but I'm just going to give them another opportunity to say succinctly as far as what they want to get out of attending the meeting.
I suppose I'll start this time. Well, here at NCBI, in terms of public data sets, we've really seen an explosion of RNA-seq data. For example, in mouse in the past 4 months, we've seen a 41% increase in the number of RNA-seq studies. So that's quite a few. And I really am very interested in what people want to do with RNA-seq and what they want to do with public data in terms of RNA-seq. There's some very exciting things coming out in terms of calling variants from RNA-seq, and I really hope to talk to people about the latest tools for calling variants from RNA-seq data. As Steve mentioned, we are also rolling out the new allele registry, and I think that's going to be something very important in terms of people interacting with our data and also through APIs. And I really want to talk to people, talk to NCBI users and see what they're interested in terms of NCBI and APIs. I also want to talk to folks about metagenomics, and to go back to some of the things Valerie was saying, I'm very interested in talking to people about structural variants in their genome, particularly unstable parts of the human genome, which are sometimes short variant calls are missed because of either structural variants or because of assembly errors. Finally, I'm excited to go and talk to the folks at the GA4GH pre-meeting and set up collaborations there.
Yeah, I have quite a laundry list of things I'm hoping to get out of ASHG this year, and obviously I'll be starting with the workshop on Tuesday. But while I'm there, I'm really looking forward to interacting with attendees. We plan on having a Q&A session, sort of a stump the chump, and so I'm looking forward to hearing from our attendees about the questions that they might have. I'm also looking forward to some follow-up meetings we have planned for groups that we've been collaborating with on NCBI and GRC related projects. I'm going to be sort of all over the place at NCBI. Not only will I be at the booth, but I'll be checking out workshops by various new technologies that are out there to stay abreast of things that NCBI should be looking for. I'm going to be going to other workshops and be looking for posters on genome tools from other groups. And I really will be out on the floor checking out posters and going to talks, particularly for people who are using the genome to see how our tools are being used in their research, and talking to them about if they're not using our tools, what could we do to help make that a possibility for them. So in general, I'm looking forward to seeing what new developments are happening out in the field, how our resources are fitting into them, and planning for new developments here at NCBI.
Thank you, Valerie, and thank you, Ben. I'm going to go back to the slides now. So you've heard a conversation amongst the panelists, and I want to underscore the fact that we will be at the NCBI booth, which is number 521, on Wednesday, Thursday, and Friday. The hall is open from 10 a.m. to 4 p.m. on Friday. The hours are or 10 a.m. to 4:15, and there will be poster sessions going on each of those days. The posters that NCBI staff are presenting were shown earlier in the webinar with their specific time. The booth is a great opportunity for you to come and have an in-depth conversation with the NCBI staff that will be there. I will be there predominantly the entire time, and then other NCBI staff will be helping me at the booth. We are located, as I said, in booth number 521. It's near the center of the exhibit area, directly across from the Agilent Technologies booth. So come and visit us at the booth. I mentioned earlier the conferences and presentations page. This is where you can get information about where we will be. The specific information about ASHG is on that page right now with a link to each of the posters. The next meeting that we will be exhibiting at is the American Society of Cell Biology in December. If you are interested in more webinars, they are listed there on the courses and webinars page. If you have a question or an idea for other webinars, please send an email message to webinars@NCBI. If you have questions about the data, tools, or web services that we provide at NCBI, please send a message to info@NCBI. If you have a specific question about your BLAST analysis, you can write directly to blast-help@NCBI. We took a poll earlier in the webinar. We had 5 choices, and it turns out that everyone agreed that we should do all 5, and that is a workshop presentation, exhibit booths, scientific talks, posters, and that is indeed, as Steve said earlier, one of the ways that we try to achieve our mission at the NCBI. Thank you for your attendance, and I hope that this was of interest to you, so we'll see you there. Thank you all.